Hello and welcome to Course 0 Lesson 2 on Model Elements. Today we'll be using two models to illustrate the main components that we use to build our model, and then we'll briefly cover the steps you need to take when you're modeling before the end of the lecture. Let's get started. Now we're going to get into some elements of a model, but first I just want to introduce you to one model that we'll be using to describe these model elements and to describe the components that you need to build a model. And the model we're going to use is the Lotka-Volterra model. This is a model of population dynamics. In it there is a predator species, here represented by a wolf, and a prey species, represented by a rabbit, and they're interacting in some way. And the way that they interact is that the predator species consumes the prey species is essentially feeding this predator species, and as a result their population numbers will vary in time. So you can imagine when the prey species, here in the blue, drops down to 0, the predator species is going to follow that because there's not enough food to feed the population, and as a result these predators will die. And so this is a cyclic behavior you're seeing here, variation over time, and that is the gist of this model. So I just want you to remember the Lotka-Volterra model as a model of population dynamics. Any model like this Lotka-Volterra model of population dynamics is going to have four main elements, and these are objects, which includes the properties and the state of the model, behaviors and interactions, initial and boundary conditions that will be specified for the model, and finally, these three model elements will then inform the dynamics and the events that we observe in our model upon simulation. Now, there are three main classes of objects, which are constants, parameters, and variables. Constants are absolute constants. These are unchangeable by the experimenter and are governed by the physical characteristics of the universe. We won't get into too much detail with these. Parameters can be changed in principle, but we usually consider them as fixed characteristics of objects. So one example of this is going to be a rate constant. A rate constant is a parameter that is very difficult to change experimentally. It is in principle possible, especially if this reaction is enzymatically regulated. Then you may be able to alter the enzyme through mutation, for example, so that it is a more effective catalyst of the reaction. But for our purposes, we're going to treat parameters as mostly fixed constants. And finally, we have variables, which can be classified into a number of different groups, but first I want to go over discrete versus continuous variables. So a discrete variable is one that can only take on some subset of the values in a given range. And a good example of this would be the number of prey or the number of predators that we have in our model. You would imagine that you might be able to have 5 prey individuals or 10 predator individuals, but you probably wouldn't see 5.5 of an animal, you know. And then next is continuous. And a continuous variable can take on any value in a range. And so you might imagine that a protein species concentration would be a continuous variable. It's going to take on some decimal point concentration within a range of reasonable physiological protein species concentrations. Now, variables can also be classified as independent or dependent variables. In the case of independent variables, these have intrinsic variability and they will not be altered by the experimenter directly. we'll focus on time and space, but you can imagine that both the Lotka-Volterra model and this biochemical model here can be modeled as time-dependent models, so that's what we'll be focusing on in this course. You could have a biochemical model that is space-dependent, so if you imagine that you have the cell as the system that you're modeling, then there might be diffusion gradients across the cell where you have some molecules in higher concentrations in one area of the cell, so therefore the concentration of that protein species will be dependent on the location it takes on. So space dependent. So these examples are time and space. And then dependent variables. These vary as a result of the independent variables, so again protein species concentrations would be variable over time, so they're dependent on the location in time. And then they also vary as a result of the changes that the experimenter makes in the model. So again, examples of dependent variables would be protein concentration, or really any biochemical species concentration. And in the Lotka-Volterra model, you can imagine it could be the number of prey individuals within the population. And finally, I just want to discuss inputs and outputs as a final classification of variables within our model. So this is pretty self-explanatory. Inputs are everything that you're feeding into your model, for example, the number of individuals in a prey population or in the predator population, and then the outputs is everything you are reading out from your model. So in the case of the Lotka-Volterra model, you would get out a certain number of predators and a certain number of prey at the end of your simulation after you've specified these inputs and run the full simulation. Additionally, in the biochemical model, you can imagine it's very similar. You input a certain concentration of a given protein species or of all the protein species in this model, and then your output will generally be the concentration of proteins within the model, and you can specify which outputs you want to look at. You can have any number of these protein species represented in your outputs. Generally, in this course, we'll be representing our output as the concentration of floating species, and outputs in this case will also be the same as the dependent variables. Objects within your model will have both behaviors and interactions. So the behaviors are what an object can do on its own, and an interaction is what objects do to each other. So here in the Lotka-Volterra model, we have as the behaviors reproduction, and death. And these are behaviors of both of these species in your model. But you can also get the interaction, which is consumption, and this is how the predator and the prey species interact. The predator consumes the prey. Now in your biochemical model over here on the right, you have production and degradation as two opposing behaviors of each protein, and then you also have interactions. So here you have activation of one protein by another, and here you have repression of one protein by another. So those are the interactions you can have in this particular biochemical model. Models also have initial and boundary conditions. So first let's go into initial conditions. These are the initial state of all model objects, so basically what our model starts out with at time 0, and usually we need to specify several of these conditions. For example, initial conditions can include parameter and variable values, which might be the concentrations of chemical species within our biochemical model, or the reaction rates. It also includes locations, numbers, and types of objects that we're representing in our model. So in the Lotka-Volterra model, we are initializing the prey species to 10 individuals in the population and the predator species to 5 individuals in the population. We would do something similar in our biochemical model. We would initialize all of these protein species concentrations. So what concentration do they have when we start our simulation? That's initializing our conditions. We would also specify that there are 2 different types of objects within our Lotka-Volterra model, and we would specify however many protein species as different types of objects within our biochemical model. I mentioned earlier that at the boundary between the surroundings and the system, you might get some edge effects where the dynamics that you observe at that boundary are different than you see within the rest of your system. We can imagine that in a Lotka-Volterra model, if something happened where, like here, both populations go to 0, and it would be very hard to rebuild a population from 0, you might have some individual of a prey population entering from the surroundings in order to create this spontaneous jump back in the population size in our Lotka-Volterra model. So that's just one example. Additionally, in our biochemical model, you might imagine that this is just some small subset of what's going on within a cell, and you might also imagine that this first protein here, the red one, is being produced in response to some extracellular signal. So even though we're not directly modeling this extracellular signal, it might have an effect on this boundary species that's coming in and basically jump-starting the entire model. And finally, we have the last of the 4 main components of a model, which are the dynamics and the events, which are represented here with 2 examples using the Lotka-Volterra model. So in the case of dynamics, these are how the states of the objects are changing continuously in time. The dynamics of a given object are completely dependent on the current states, configurations, behaviors, and interactions in which the model is participating. So here we see oscillations occurring over time in both the predator and the prey species. And these are dependent on the initial conditions of the predator and prey populations, as well as how these species interact with one another and the rate at which they reproduce and die naturally, which is a behavior intrinsic to each of the species. We also have events, which are changes in the model state that are imposed from outside of the model, and they occur discontinuously in time or space. Again, we'll be using time primarily, and this means that there's a clear point of initiation or termination of whatever the event is that occurs. So here we have a point at which the predator species completely die. Let's imagine, for example, that the predators are struck by some disease that killed the population and it's unable to build itself back up. In this case, we have this strange behavior occurring in the prey output where it just continues to rise over time, and it looks like it's becoming this massive prey value because there's no predators to keep the population of prey in check. So again, this happens at just one specific point in time. Otherwise, we had these oscillatory dynamics as expected before we had this event that completely changed the output of our system. I want to very briefly touch on the ideas of deterministic and stochastic models. So, deterministic models, which we'll be primarily using in this course, they always produce the same output for a given input. Then we simulate the Lotka-Volterra model and we get this oscillatory output that we've observed before, and then we go ahead and we reset the model to all the same initial conditions, all the same initial assumptions, and we simulate it again. We get the same deterministic output. We get the same oscillatory dynamics given those initial conditions. In the case of a stochastic model, it's not true that you'll get the same output given those same inputs. So imagine that we have some element within the Lotka-Volterra model that introduces randomness into our output, despite having the same input. In this case, when we simulate the model, we might get some output that looks like this. So here you see that the predator species initially rises in response to the prey species, but after the prey species drops down to 0 and the predator species falls, neither one of these populations can rise back up for the rest of the time course. So that's one output you get from simulating this model. Well then, you reset the model to the same initial conditions and you simulate again, and in this case, for some reason, the predator species dies in the model, and it cannot rebuild its population, and as a result the prey species just proceeds to an infinite number in the case of the way these mathematics work out. So this is considered stochastic. Now, to kind of understand stochasticity, you might imagine that a coin flip is governing some element of the model. So say that every time a wolf encounters a rabbit, that you flip a coin to determine whether the wolf is able to consume the rabbit. So let's say you get heads and the rabbit is consumed by the wolf. But let's say instead you flip the coin and you get tails, and the rabbit is not consumed by the wolf, and as a result this will affect how the predator species responds, and it's completely random because it's just governed by something like a coin flip. Alright, now that we've gone through what a system is, what a model is, and what elements you need to create your model, what are the actual steps you need to take to build your model? Well, the first step is to use existing experimental data to inform your model, and this will feed into the second step, which is defining your conceptual model. To do this, you define your system objects based on elements you know exist and are relevant to your model from experimental data, you define your system boundaries, and you define your simplifying assumptions. You will then go ahead and define your quantitative model template, which is invoking physical laws that will describe your system processes. And then you define your mathematical model, which will determine whether processes are going to be modeled stochastically or deterministically, and will also determine whether objects, usually in our case this will be biochemical species, will be modeled as discrete individuals or as continuous concentrations. And then the last step is to test your model against experimental data and to go ahead and repeat this process until you are confident that your model is the most accurate representation of the experimental knowledge that you have, and that it is capable of making accurate predictions.
And that concludes Lesson 2, Model Elements. In our next lesson we will be discussing cellular networks so that you can better understand what this visual representation of a biochemical model actually means.